from beginning again. Okay. Principles and applications of genetic recombinant technology and strain improvement of industrially important organisms. Mutational rDNA technology. Various approaches used to achieve strain improvement of industrially important organisms. Selection, mutation, manipulation of biochemical pathways using analog, inhibitors, inducers, regulatory control, and of course, you have something called as overproduction, which means higher amount of production producing strains. Now, when we come into the topic, we equally have modern genetic techniques that can bring about what is called as mutations in production strains. So modern genetic techniques are now used. Recombinant DNA technology is not only used for quantitative yields, but also for the qualitative yields. So recombinant DNA technology doesn't only yield the amount of product in a higher rate, but the quality equally is maintained. So rDNA technology is not only used for quantitative yields, but also for the qualitative yields. In any strain improvement efforts, rDNA technology will be useful if any one or few genes are capable of affecting the improvement, but where complex pathways are in question, the successes are difficult. I tell you, or I explain you in this way. So recombinant DNA technology is possible only when there are few genes that is responsible for production of a product. If it is in a multi-enzyme system or where complex pathways are involved, the success rate of using recombinant DNA technology is not feasible. So I repeat, in any strain improvement efforts, recombinant DNA technology will be useful if any one or few genes are capable of affecting the improvement, but where complex pathways are in question, the successes are difficult. If key enzymes on pathways with their activity are well understood, then rDNA technology has more scope. So what is the gene responsible for the enzyme and what is the enzyme responsible for the production? If you understand that in a neat manner, then rDNA technology is highly applicable, is what this sentence means. I repeat, modern genetic techniques are now used. rDNA technology is not only used for quantitative yields, but also for the qualitative yields. In any strain improvement efforts, rDNA technology will be useful if any one or few genes are capable of affecting the improvement, but where complex pathways are in question, the success rate is difficult. If key enzymes on pathways with their activity are understood, then rDNA technology has more scope. Why strain improvement? So why do we require strain improvement? If you see, the main objective of any strain improvement procedure is the economy. So economy is the overall objective of industrial production. So profit is one big thing that we require. Commercial exploitation of microorganisms will be successful if it competes alternate chemical synthesis methods on all fronts. So you need to exploit microorganisms by altering its chemical pathway synthesis, so it may be successful in all fronts. Applications of gene technology to improve industrial strains are found for different product classes, including the production of antibiotics, vitamins, enzymes, amino acids, alcohols, etc. Reduced production cost, improved rate of synthesis, widening of substrate utilization, and emerging of new production organism could be the various means of achieving ultimate economy. Okay, maybe I can make you easier this way. Why is strain improvement rather than reading the slides as such? So strain improvement via rDNA technology or through gene technology is very much used for industrial strain, and it is used for producing various classes of substrate from the microorganisms, starting from antibiotics, vitamins, enzymes, amino acid, and alcohol. The main objective of any strain improvement process may be genetic or non-genetic, if you see, or altering chemical pathways because of the economic improvement or because of economical development. And how are we going to ultimately achieve this economy, if you see, is reduction of production cost. So production cost romba adhigama irundhuchu appadina manufacturing cost adhigama poidum. So production cost, how far we can reduce. So reduction of production cost, improved rate of synthesis. How are we going to improve the synthesis of the product by the microorganism, and widening of substrate utilization. I think you studied something called as production medium when you went across antibiotic production. So we studied about carbon source, nitrogen source, and various mineral sources. How far you widen the microorganisms' role in utilizing the substrate. And of course, emerging of new production organisms, finding out newer organisms to produce various products could be the means of achieving ultimate economy. So I repeat the slide. Economy is the overall objective of industrial production. Commercial exploitation of microorganisms will be successful if it competes alternate chemical synthesis methods on all fronts. Applications of gene technology to improve industrial strains are found for different product classes, including antibiotics, vitamins, enzymes, amino acids, alcohols, etc. Reduced production cost, improved rate of synthesis, widening of substrate utilization, and emerging of new production organism could be the various means of achieving ultimate economy. So what are the overall objectives of strain improvement? So you have various procedures, and why do we bring about, or what is the objectives, or how are we going to bring about strain improvement? It is a very important 5-mark question as such. The main objective of strain improvement, if you see, is to produce strains which gives you higher yields. Strains giving higher yields. Number 2 is strains requiring shorter fermentation time. So when you studied about production of antibiotic penicillin, the production time is 4 days. You cannot wait months together investing on something. So you should have a strain that requires shorter fermentation time. Strains producing no desirable pigments. So if they produce undesirable pigments, the production will ultimately go waste. So strain should not produce any pigments. Strains having reduced oxygen demand. So submerged fermentations are there. So you need to supply aeration for it. Sterile air you need to supply in vessel volume per minute. So strains that can use reduced amount of oxygen so that it can give you an effective end product. So oxygen, as you supply aeration inside and fermenter vessel, the ultimate difficulty we have is the generation of foam. To control that, if the strain could produce an effective end products with the reduced oxygen demand, that would be of utmost use. Strains with decreased foaming. Strains using inexpensive substrate or amply available substrate, which means you're going to produce something, you're investing on something. Imagine a situation if the organism uses very costly means for its development. It is very difficult for you to produce something. So either the substrate that the organism uses should be amply available. It should be an inexpensive substrate. So strains using inexpensive substrate or amply available substrate. Strains not producing mixture of related compounds but producing single metabolite of interest. So in case if it is biochemically producing a mixture of compound, recovery, downstream processing, so that will become very difficult. So recovery of the product, the procedure of recovery of the product will be difficult. So strains should not produce mixture of related compounds, but it should produce single metabolite of our interest. Strains aiding in easy product recovery, of course. Strain giving more effective end product. The product that it gives should have its purpose. So strains giving more effective end products. Strains giving products with reduced toxicity. This is very applicable for any pharmaceuticals like vaccines, antibiotics, or anything that we consume. For alcohol, whatever we take as a food product also. In case if it is toxic, we can no way use it. So strains should give only a very reduced toxicity. Strains giving product with improved activity. That is the ultimate aim. So it should produce an improved activity, and strains giving newer products, varieties of products it should generate. Okay, so I repeat the slide. Objectives of strain improvement: strains giving higher yields, strain requiring shorter fermentation time, strains producing no desirable pigments, strains having reduced oxygen demand, strains with decreased foaming, strains using inexpensive substrates or amply available substrates, strains not producing mixture of related compounds but producing single metabolite of interest, strains aiding in easy product recovery, strains giving more effective end product, strains giving products with reduced toxicity, strains giving product with improved activity, and of course strain giving newer products. So these are the objectives of strain improvement. So there are a few examples of achievements that has been carried out using rDNA technology in improving strain. The first one is plasmid transfer. Gene manipulation can also be achieved by plasmid transfer, which was done in a strain Streptomyces collicolor. So there are two plasmids, PUJ2303, that produces an antibiotic granitizin, and AM7161 medermizin. These two are cloned together to produce a plasmid that can produce granitizin and medermizin. So gene manipulation can be achieved by gene transfer. Production of antibiotic. One example I've given you is semi-synthetic penicillin. Production of recombinant insulin, recombinant vaccine, and of course growth hormones for dwarfism, single cell protein, microbiological pesticides, and of course you have genetically engineered microorganisms or genetically modified microorganisms, gems அப்படின்னு சொல்லுவோம், to degrade xenobiotic compounds, the compounds that cannot be easily degraded, or xenobiotic or recalcitrant compound அப்படின்னு சொல்லுவோம், recalcitrant chemical அப்படின்னு சொல்லுவோம். அந்த மாதிரி இருக்கக்கூடிய உடைக்க முடியாத காம்பவுண்டை உடைக்கிறதுக்கு we use genetically modified or genetically engineered microorganisms, which you shortly or cutely call them as gems. So I repeat the examples of achievement: gene manipulation by plasmid transfer, production of antibiotics, semi-synthetic penicillin, recombinant insulin, recombinant vaccine, growth hormone dwarfism, single cell protein, microbial pesticide, and gems. And of course, more than this, you have very many varieties. In plant resistant varieties நிறைய இருக்கு. Tissue plasminogen activator ne unga syllabus la irukku, interferons irukku. So everything is one achievement of what is called as rDNA technology in production of improved strain. So the next slide will tell you what are the strategies of strain improvement. So when you have a producer strain, how are we going to make it an high yielding variety? What are the strategies that you will follow in improving the production strain? So producer strain, you will identify the key enzyme that is responsible for production pathway. So the first one is you should identify the key enzyme causing the bottleneck in the production pathway. Identify the physiological characteristics of different producers. So each organism will have a different need for its growth. Okay, so one may require a higher amount of carbon source, one may require a higher amount of oxygen supply, one may require a higher amount of micro and macro elements. So it depends. So you will identify the physiological characteristics of different producers. And you will also identify the gene responsible for specific metabolic activity. Example, carbohydrate substrate utilization. So I repeat, when you have a producer strain, you should identify the key enzyme that is causing the bottleneck in the production pathway. You will identify the physiological characteristic of different producers. You will identify the gene for specific metabolic activity. So what you will do later is you will isolate the respective gene. Without gene isolation, you cannot go in for gene cloning. So you have to go in for isolation of respective genes. You will note the deficiencies and qualities required for the supply in a medium for the production strain. So in case if it cannot produce any vitamins, or it cannot give rise to any minerals, you will supply that in addition. So you will note the deficiencies and qualities required for its physiological characteristic, and it will be cloned in an weaker producers incapable of using that substrate. For example, insulin is produced using Escherichia coli. E. coli nowhere produces insulin naturally. It is because of gene cloning. Even though it is not a producer, you're cloning it inside an Escherichia coli cell to produce an end product. So the second stage which you will bring about is isolation of respective genes, note deficiencies and qualities, cloned into weak producers, which is incapable of using the substrate. After cloning the respective genes, you have to go in for increasing its copy number. I think you all studied copy number when I taught you plasmids. So in case if the plasmid is an stringent plasmid, you have to convert it to an high copy number plasmid. The only reason is to produce overproduction of the product. So in case if you want to produce in a high quantity, high yielding product, you have to go in for the increase of copy number of the gene. You can do protoplast fusion to combine qualities. If you have two different cells with two different requirements, you can fuse them together to give supplementation of deficiencies, and that is what you call it as hybridoma technology or hybrid technology. So protoplast fusion can be carried out. And expand the raw materials utilization. You can supply the raw materials in a higher quantity, or you can expand the raw material utilization. And if you carry out all these things, if you carry out all these procedure, you will definitely go in for an improved strain that you can get. So you can obtain high yielding varieties or improved strains. So I repeat, strategies of strain improvement: producer strain, identify key enzyme causing bottleneck in production pathway, identify physiological characteristic of different producers, gene for specific metabolic activity, example carbohydrate substrate utilization, isolation of respective genes, note deficiencies and qualities, cloned into weak producers incapable of using that substrate, cloning of genes and increasing the copy number, protoplast fusion to combine qualities, supplement on deficiencies which are hybrids, and expand raw materials, carbohydrate substrate utilization to improve the production strain, or to obtain what is called as an improved strain. Okay. So now we are going inside mutational rDNA technology. So molecular biology, the paper, the concept is playing with genes. So we are going with mutational rDNA technology, and those definitions that will come now, if you see, those are all 2 mark of importance. So you can also write down if possible, or note down when I tell you things. A technique mainly used to change the phenotype of an organism. So here what we are talking is about the host strain or the host organism. When a genetically altered vector is introduced and integrated into the genome of the organism, this gene which is introduced is called the recombinant gene, and the technique is called as recombinant DNA technology. So you are changing the genotype of an organism, so it is phenotypically expressed. A technique mainly used to change the phenotype of an organism. When a genetically altered vector is introduced and integrated into the genome of the organism, this gene which is introduced is the recombinant gene, and the technique is called as recombinant DNA technology. Now, what is in vitro mutagenesis? So the word in vitro mutagenesis, the word in vitro is away from the body, in vivo within the body. So mutation brought out in a laboratory condition is in vitro mutagenesis. More simplified, you cannot tell. So any mutation carried out away from an organism, host organism, in the laboratory condition, you call it as in vitro mutagenesis. Techniques whereby DNA mutations are deliberately engineered to produce mutant genes, proteins, or strains of organisms. In vitro mutagenesis is used specifically to change the base sequence of a segment of DNA, which may either be general, random, or targeted, and they are also referred to as protein engineering. I repeat, technique whereby DNA mutations are deliberately engineered to produce mutant genes, proteins, or strains of organism. In vitro mutagenesis is used specifically to change the base sequence of a segment of DNA, general, random, or targeted, and they are also referred to as protein engineering. We will go again with mutational rDNA technology, a technique mainly used to change the phenotype of an organism when a genetically altered vector is introduced and integrated into the genome of the organism. This gene, which is introduced, is the recombinant gene, and the technique is called the recombinant DNA technology. Technique whereby DNA mutations are deliberately engineered to produce mutant genes, proteins, or strains of organisms. In vitro mutagenesis is used specifically to change the base sequence of a segment of DNA, which may either be general, random, or targeted, and they are also referred to as protein engineering. So mutational rDNA technology, or what you call as in vitro mutagenesis in the laboratory condition, can be brought out by two methods. One is site-directed mutagenesis, or it is also called as oligonucleotide-directed mutagenesis. The other procedure we have is cassette mutagenesis. I repeat, mutational rDNA technology or in vitro mutagenesis can be brought out by two means. Either by site-directed mutagenesis. Site-directed mutagenesis is also referred to as oligonucleotide-directed mutagenesis. On the other hand, we have something called as cassette mutagenesis. And these both procedures or techniques are employed to bring about mutation in increasing the yield of the production strain by using mutational rDNA.